Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Nature uses 20 canonical amino acids as building blocks to make proteins, combining their sequences to create complex molecules that perform biological functions. That's the terrain Princeton University's Hecht lab works in. And recently, their curiosity for designing their own sequences paid off. They discovered the first known de novo protein that catalyzes, or drives, the synthesis of quantum dots. Quantum dots are fluorescent nanocrystals used in electronic applications from LED screens to solar panels. Their work opens the door to making nanomaterials in a more sustainable way by demonstrating that protein sequences not derived from nature can be used to synthesize functional materials, with pronounced benefits to the environment. Quantum dots are normally made in industrial settings with high temperatures and toxic, expensive solvents, a process that is neither economical nor environmentally friendly. We're interested in making life molecules, proteins, that did not arise in life, said Professor of Chemistry Michael Hecht, who led the research with Greg Scholes, the William S. Todd Professor of Chemistry and Chair of the department. In some ways we're asking, are there alternatives to life as we know it? All life on Earth arose from common ancestry. But if we make lifelike molecules that did not arise from common ancestry, can they do cool stuff? So here, we're making novel proteins that never arose in life doing things that don't exist in life. The team's process can also tune nanoparticle size, which determines the color quantum dots glow, or fluoresce, in. That holds possibilities for tagging molecules within a biological system, like staining cancer cells in vivo. They're very good at absorbing light and converting it to chemical energy. That makes them useful for being made into solar panels or any sort of photosensor. But on the other hand, they're also very good at emitting light at a certain desired wavelength, which makes them suitable for making LED screens. And because they're small, comprised of only about 100 atoms and maybe 2 nanometers across, they're able to penetrate some biological barriers, Maki. The research, a de novo protein catalyzes the synthesis of semiconductor quantum dots, was published this week in the Proceedings of the National Academy of Sciences, PNAS. A key word for me is engineering. I want to be able to engineer proteins to do something specific, and this is a type of protein you can do that with. We can achieve better quality by engineering the protein to influence quantum dot formation in different ways. Based on work done by Sarangan Chari, Hecht Lab's senior chemist and a corresponding author, the team used a de novo protein it designed named conc to catalyze the reaction. Researchers first isolated conc in 2016 from a large combinatorial library of proteins. It's still made of natural amino acids, but it qualifies as de novo because its sequence doesn't have any similarity to a natural protein. Researchers found that conc enabled the survival of E. coli in otherwise toxic concentrations of copper, suggesting it might be useful for metal binding and sequestration. The result is CD's quantum dots. To make a cadmium sulfide quantum dot, you need the cadmium source and the sulfur source to react in solution, said Spangler. What the protein does is make the sulfur source slowly over time. The research was also supported by NSF grant MCB 1,947,720 to MH. Materials provided by Princeton University. Original written by Wendy Plump. Note: content may be edited for style and length. Princeton University. 2022, December 13. Quantum dots at room temp, using lab-designed protein. Science Daily. Retrieved December 13, 2022 from ift.tt/mg4p78 Princeton University. Thanks for watching. Please subscribe my channel for more videos.